This presentation reviews the genetic basis of hydrocephalus, using the 2024 article by Hale and colleagues as the main source. The central idea is simple. Hydrocephalus is not one genetic disease. It is a shared clinical endpoint that can arise from disrupted brain development, cilia and ependymal biology, cerebrospinal fluid regulation, extracellular matrix integrity, and injury susceptibility. Why bring genetics into a condition we usually treat surgically? Because the same visible endpoint, enlarged ventricles or abnormal cerebrospinal fluid dynamics, can come from many upstream causes. Congenital, syndromic, post-hemorrhagic, post-infectious, tumor-related, chiari-associated, neural tube defect-associated, and adult normal pressure forms overlap clinically. Shunts and endoscopic third ventriculostomy remain essential. But genetics may help explain risk, prognosis, recurrence, and targets beyond diversion. Hale and colleagues set out to organize a fragmented literature. Their question was, what genes, pathways, and mechanisms have been reported to contribute to human hydrocephalus, and where are the evidence gaps? The review is phenotype first, then mechanism focused. It does not treat every reported gene as proven causal. Instead, it asks how reported genes cluster into biological themes, and how pleiotropy connects hydrocephalus with other central nervous system and non-central nervous system features. The methods matter because this is a broad evidence map, not a single cohort study. The authors search PubMed and OMIM using hydrocephalus ventriculomegaly and genetic descriptors. They identified 3,804 records, removed 95 duplicates, screened 3,709 records, and performed full-text screening on 1,057. After eligibility review, 327 studies were included. Those studies vary widely. Case reports, sequencing studies, COPA number findings, cohort analyses, and pathway observations shape the synthesis. Figure 1 presents an overly narrow view of genetics. A variant may cause a primary malformation, disturb cilia or ependymal cells, alter cerebrospinal fluid secretion or absorption, or increase vulnerability to hemorrhage or infection. So the genetic question is often not which hydrocephalus gene is present. A better question is, what else is this gene doing and in which developmental window or tissue? This slide is the roadmap for the mechanism sections. The review groups evidence around aqueductal stenosis, X-linked and L1CAM-associated hydrocephalus, ciliopathy and malformation syndromes, signaling pathways, extracellular matrix and barrier mechanisms, neurogenesis, adult normal pressure hydrocephalus, and broader regulatory or metabolic pathways. The caution is that this is a map of reported associations, not a ranked list of causality, and it still needs human-specific validation. Aqueductal stenosis can sound like a mechanical obstruction, but the genetic findings point to broader biology. The review reports 15 unique gene mutations on 11 chromosomes in human hydrocephalus secondary to aqueductal stenosis. Representative genes include L1CAM, AT1A3, SLC12A6, FGFR3, CRB2, MPDZ, LAM1, ADAMTSL2, COL4A2, and SOX2. Mechanistically, these implicate ion transport, brainstem development, cell polarity, extracellular matrix structure, and choroid plexus biology. L1CAM is the classic example of locus complexity and pleiotropy. The article summarizes missense, nonsense, insertion, duplication, deletion, microdeletion, and splice region events across the gene. The phenotype extends beyond ventricles. L1CAM-associated hydrocephalus overlaps with Massa syndrome, adducted thumbs, spastic paraplegia, callosal abnormalities, and neurodevelopmental impairment. The teaching point is that one locus can produce a spectrum, not a single tidy hydrocephalus phenotype. Cilia genes bridge molecular biology and cerebrospinal fluid movement. Genes such as MKS family genes, IFT genes, CEP83, FOXG1, DNI2, and WDR16 point to intraflagellar transport, motile cilia function, vesicular docking, and apical polarization. Dandy-Walker and posterior fossa genes add another layer. Abnormal cerebellar and posterior fossa development may alter fluid pathways, but it also reflects deeper developmental patterning rather than a simple obstruction alone. The gene lists become more meaningful when we look for convergence. Pi3K ACTM-TOR genes, WNT and planar polarity genes, and FGFR or TGF beta-related pathways regulate growth, patterning, epithelial polarity, and tissue mechanics. That means pleiotropy is expected. The same pathway can shape brain growth, skull formation, cilia organization, ependymal integrity, and extracellular matrix behavior. Hydrocephalus can be one output of a broader developmental signaling disturbance. This group of mechanisms is clinically intuitive. Hydrocephalus depends on the tissues that contain, move, and absorb cerebrospinal fluid. Vesicle trafficking, tight junction integrity, dystroglycan glycosylation, basement membrane stability, collagen and fibrillin networks, and water or ion regulation can affect that interface. Genes such as MPDZ, POMT1, POMGT1, COL4A1, LAM1, ACT4, AQP1 regulators, and TRPV3 fit a tissue interface model of pathogenesis. One important shift in the review is away from a purely hydrodynamic model. Many reported genes point to neural stem cell fate, neuroepithelial integrity, cortical development, planar cell polarity, and neuronal migration. Examples include TRIM71, SMARC1, SOX9, PTCH1, ADGAG1, TUB3, SCRIB1, PTK7, VANGL2, and DVL2. Clinically, this matters because poor neurodevelopmental outcome may reflect the primary developmental disorder, not only delayed or ineffective cerebrospinal fluid diversion. The remaining categories are diverse but not random. Transcriptional and epigenetic regulators, metabolic genes, cell cycle genes, cytoskeletal and cytoarchitral genes, and lipid regulatory pathways recur in the literature. These mechanisms may influence resilience of developing neural tissue, epithelial organization, protein processing, and cellular structure. The practical takeaway is that hydrocephalus genetics is not just a list of fluid genes. It is a window into developmental regulation and tissue stability. The chromosomal map reinforces the same message visually. Reported hydrocephalus-associated loci are spread across the genome, including autosomes and sex chromosomes. This argues against a single-region model and supports genetic heterogeneity, with contributions from kappa number variants, rare single-gene disorders, inherited variants, de novo variants, and non-coding regulation. For the audience, the figure is less about memorizing locations and more about appreciating the breadth of the biology. Normal pressure hydrocephalus has less genetic evidence than congenital and syndromic forms, but the signals are still interesting. Reported associations include SFMBT1, expressed in ependymal and epithelial cells, CFAP43 with cilia and flagella-related biology, the ETNF locus on chromosome 19q, and CWH43 deletion. These findings may eventually separate communicating hydrocephalus into ependymal, ciliary, absorptive, vascular, or neurodegenerative overlap subgroups, but the evidence remains early. The global representation gap is one of the review's strongest messages. Genetic studies are concentrated in regions where hydrocephalus burden is comparatively lower. The authors note the absence of studies from sub-Saharan Africa, despite high disease burden and high genomic diversity. This is both an equity problem and a scientific problem. Without diverse cohorts, counseling tools, risk prediction, and future precision therapies may be incomplete. This synthesis slide puts the pieces together. Genotype can influence developmental programs including neural stem cell fate, skull and posterior fossa patterning, and epithelial polarity. It can also affect the tissue and cerebrospinal fluid interface, ependymal cilia, choroid plexus, extracellular matrix, barrier integrity, and water or ion transport. The clinical phenotype is downstream. The useful questions are which tissue, which window, and is fluid dysregulation primary, secondary, or both? The translational message should be cautious. Genetics is not ready to dictate most hydrocephalus treatment decisions, but it can reshape diagnostic thinking, genetic counseling, recurrence risk assessment, family planning, extracentral nervous system surveillance, and prognosis. It may also help separate pressure-related morbidity from primary neurodevelopmental disease. In the future, genotype outcome links could inform shunt, endoscopic third ventriculostomy, or choroid plexus cauterization counseling and support pathway-targeted trials. The strength of this review is that it imposes structure on a fragmented field. It integrates phenotype, genes, pathways, chromosomal location, and global representation. The limitations come from the underlying literature: small studies, case reports, inconsistent phenotyping, publication bias toward rare and syndromic disease, and variable evidence for causality. The best use of this paper is as a hypothesis-generating framework, not as a definitive clinical gene list. The research agenda follows naturally. The field needs deeper phenotyping, standardized imaging and outcome data, diverse high-burden cohorts, whole exome and whole genome sequencing, kappa number detection, multiomics, and human validation through tissue organoids and induced pluripotent stem cell models. Most importantly, genetic data should be linked to outcomes that matter. surgical response, complications, cognition, epilepsy, survival, and quality of life. The goal is variant to mechanism to phenotype to intervention. The take-home message is that hydrocephalus is a heterogeneous final phenotype, not a single pathway disease. Genetic findings converge on development, cilia and ependymal biology, cerebrospinal fluid regulation, extracellular matrix and barrier integrity, and cell signaling. Current evidence is broad but uneven, so causality and clinical utility require better phenotyping and human-specific validation. The largest opportunity is also an equity imperative. Diverse cohorts linked to longitudinal outcomes.